Okay, sir. Good afternoon, guys. Today I'm going to talk about the structure of DNA. DNA stands for deoxyribose nucleic acid, and DNA is a molecule which is located inside the nucleus of the cell. Located inside the nucleus of the cell, and its main job is to store and code the genetic information. Mostly in the life cycle of the cell, the DNA is located inside the nucleus of the cell like coiled noodle. Now I show you in the picture. It's like coiled noodle, but during the time of the cell replication, the DNA is arranged in the form of the structure which are known as chromosome. And the chromosome helps to keep the DNA stable during the time of the cell replication. Most of are familiar with the double helix model of the DNA. The double helix DNA demonstrates the DNA is basically a polymer. To understand the structure of the DNA, you have to understand the definition of the polymer. Polymer is a molecule consisting of many repeating units which are known as monomers. If we taken as an example, polymers called as starch. In this case, the repeating monomeric units are known as glucose. These glucose molecules are joined together like glucosidic bonds, and it forms the polymer which is known as starch. In a similar way, the DNA is also a polymer, and in case the monomeric unit which is in the form of polymer DNA are known as nucleotides. DNA is a nucleotide. You can see that it consists of three important groups. The phosphate, the sugar, and the nitrogenous base. The sugar present inside the nucleotide is deoxyribose, which is 5th carbon sugar, which belongs to the pentoses. The fossil group consists of phosphorus ion, to which 4 oxygen atoms are attached, and then we have important nitrogenous bases which are organic molecule which consist of nitrogen in the form of ringed structure. We have 4 different types of bases that can be present inside the bases. They are adenine, guanine, cytosine, thymine. Now, since we can have 4 different types of nitrogenous bases that are present inside the structure of DNA. Now you can see the nucleotide are arranged in the 2 groups. The first group are known as paramidins, and these consist the nitrogenous bases thymine and cytosine, which are both single ringed bases. And the second group consists of nucleotides purine, which is adenine, and guanine as the nitrogenous bases, which are double ringed in structure. Now in case of starch, the structure which only consists a long luminar chain of the glucose or either branched chain which are joined together by glucosilic bonds. This is the chemical structure of starch. But in the case of DNA, the 4 types of nucleotide join together in the complex way from the double helix structure. The blue vertical line basically represented the sugar phosphate backbone, and the central organic horizontal line represent the base pair of the DNA. In this picture, In this picture, the blue vertical line basically present is the sugar phosphate, is the backbone, and the central orange horizontal lines is represented the base pairs of the DNA. How two nucleotides are joined together. The carbon present inside the deoxyribose sugar of the DNA. The carbon present inside the nucleotide in the clockwise direction are presented in 1 to 5 clockwise direction of the DNA. The 3 carbon is every deoxyribose sugar of bond with the phosphate group. The 3rd carbon in every deoxyribose is from the phosphate group, and another nucleotide present below it, and this bonding extends in the both direction. And this bonding extends in both direction. Every phosphate is connected to the 2 sugar, one bond with the 3rd carbon sugar with up of, and one bond with the 5th carbon sugar below. In this picture, the one phosphate is connected to the sugar in the upward direction, and one is connected with the downward direction. One is bond with the 3rd carbon sugar, and another one is bond with the 5th carbon sugar. Due to this bonding, the formation between the sugar and the phosphate, it creates the sugar phosphate backbone. Due to this bonding, the backbone will be created one side, and we have all the nitrogenous bases with project out the sugar phosphate backbone on the sides. Due to this bonding, at the one side, it will be project the backbone. So it is basically represent, it is the single strand of DNA. Now we can clearly understand. To understand the key difference, first we have to understand the direction of the complementary base pairs. To understand this, we have to clearly understand about this complementary base pair which is created between the 5 and 3. Direction means it is the end-to-end chemical orientation of single-strand nucleotide acid in single-strand nucleotide acid. For example, we can see the single-strand nucleic acid. The direction of DNA is expressed the direction of the DNA is expressed in 2 ways. Chemical orientation of single strand of nucleic acid. In 5' end, 5 phosphate group is attached. In 5' end, the 5 phosphate end is attached. In 3' end, hydroxy group is attached. In 3' end, the hydroxy group is attached. If we come back to the DNA ladder model, we can see the anti-parallel. If we come back to the DNA ladder model, we can see the anti-parallel. The left side is the 5' to 3', and the opposite side is 3' to 5'. So it is known as anti-parallel. The next important line to understand is complementary base pairing. Complementary base pairing decreases the manner in whom the nitrogenous base of the DNA molecule aligned with the other. DNA molecule is aligned with the other, so it is basically mean that base adenine pair always with thymine and base guanine always the adenine is always with thymine and cytokinin is always with guanine. So it makes the hydrogen bond with each other. We see this in the DNA ladder model. It is very important for the stability of the two strand of DNA. It is so important for the stability to transform for longer period. Now we can understand the double helix structure of the DNA. It is a double helix structure of the DNA. This is the function and structure of the DNA. Thank you, sir.
What is that